meant to have it already open. So which portion, A, B, or C, do we call the FC region of an antibody? A, B, or C? Which one has the FC region? Which one? C, right? It wasn't a trick question, y'all, honest. All right. So therefore, what's the function of parts A and B? What do these two parts do? What do A and B do? What do antibodies do? Attack. By doing what?
Attaching
to the antigens, right? So they actually attach to the antigen. So these are our antigen binding fractions, remember? A, B for antigen binding. So these are the two pieces that attach to the antigen. This is the piece, right, that stays constant, that our phagocytic cells bind to. So what part of each antibody is different is the variable region. It is the green or the blue here? The green. The green is what changes. Near the antigen binding sites is your variable region, right? Your blue is what's constant, what stays the same, right? From class to class of the antibodies, so that the immune system can recognize the type of antibody and so that this structure forms. All right. So D, remember we don't know much about this antibody. It's very rarely secreted. But I was doing some reading today, and it actually is found. It can be found on the surface of the B cell. In addition to which class of antibody do we say is the main receptor on the surface of the B cell? IgM. You got it. You got it. Okay. And then E was kind of a specialist too, right? What does it bind to? It's on the slide. Right, but how does it bind to it? What part of the antibody binds to the mast cell in the basophils? A and B is for antigen binding, right? It's the FC region, right? So a lot of times these receptors are referred to as FC receptors, but in this picture it's saying IgE receptor because it's specifically referring to just that class of antibody can bind to mast cells and basophils. Okay, and then we talked about three others: IgM, IgG, and IgA. IgM, as we've already said, that one is in this monomer form. It is the B cell receptor, basically. Which one is this big pentamer when it's secreted by the B cells? IgM, right? Remember, mega big. Okay, and then when it goes into our secretions, it's a dimer. And which one is that? IgA. And what do we mean by secretions? What's an example of a secretion that this would be found in?
Breast milk, mucus, and tears.
Breast milk, mucus, tears. You forgot one.
Saliva.
Saliva. Saliva. Okay. So these antibodies we already know bind to specific antigens, right? But how do they help us by binding, right? So the main ones that do a lot of these outcomes, right, have a lot of these things happen, are our IgA, whether it's in its monomer form in our blood or if it's in our secretions as that dimer. IgG, which is always that monomer, which IgM is that big pentamer we just looked at when it's secreted, right, really big. So tagging for destruction, do you think IgA, IgG, and IgM could do that? Actually tag it, opsonize it, and then a phagocytic cell recognize that and engulf it? Yes. Oops, I started in the wrong spot. Should have numbered my things. Okay, so I already gave you the answer. Can they neutralize? Can they bind to viruses and stop them from attaching to us? Can they stop toxins from working? Yes, right? All three of these can do that. What about immobilizing them, wrapping up their flagella, or stopping them from adhering to us, like these modal bacteria here? Can IgA do that? What you think? Yeah? What about IgG? All three of these can do this functionality, right? They can tie up the flagella, immobilize them that way. They can bind to them and therefore stop them from adhering to us. Would IgG, IgM, and IgA be good at cross-linking, agglutinating, and precipitating different antigens? Yeah. Who do you think would be the best at really causing a whole bunch of cross-linking? IgM, why? Because it's big. It has how many antigen binding sites on it? It's 5 antibodies. Each antibody has 2, so that's a potential of 10 things that one antibody could bind to. Right? Although they're showing the monomer here, right? IgM can do this as well. And I'm going to try and find us some pictures of that. Now, 2 of these outcomes are specialist, right? For this outcome of an antibody binding and a natural killer cell having a receptor, an Fc receptor for that specific antibody, and killing that cell because of the antibodies detecting something's wrong with that cell, this process of antibody-dependent cellular cytotoxicity, there's only one class of antibody could do this. Which one of the 5 is this? Anyone know? What's our most abundant one in our body? The longest lived antibody?
IgG.
Yep, IgG. Can cross the placenta, right? So, complement activation, right? So, when an antibody binds, anyone know what activation that is called? Is it the alternative, classical, or lictin binding activation when an antibody binds? And it is the classical. And there's 2 of them that can initiate activation of the complement system. Which 2? What are our 2 main ones? What's the first one we produce? Even when we're a fetus, we can make this one, IgM, right? And what's our main one for protection all throughout our body? The IgG. So it's both IgM and IgG. Does that make sense to you guys? Yeah. Both of these can activate complement. So, think about it, right? We introduced it, right? And so we just kind of layered it together, right? What's the significance? What can these things do? So today we're going to look at this process, right, of the cells actually being selected, activated when they've found an antigen with their receptors, whether it be a B cell or a T cell. So you'll notice that I highlighted T cells here because although picture-wise and I think this animation-wise, we're going to focus on B cells, this same process happens for T cells. They have to become activated before we're going to produce more of them. And the other key thing to remember is that each B cell or T cell receptor, on that B cell, they're all the same. On that T cell, they're all the same. They all recognize the same antigen, okay? All the receptors, even though there's more than one, they all recognize the same thing. And each B cell or T cell has made its own very special receptor. It's different from any other B cell you see or any other T cell. Right? Remember, this is a high level of specificity that happens here. So I want you to remember that, and I'm gonna play through this animation, and I'm gonna try and hold up my headset so hopefully it records. But of course, you guys have links and everything to these things, so you can play them again if you want to. and then I'll pick through it.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
Okay. So let me hit pause so that it'll not play on me. I just want the pictures. So remember, all our blood cells, including the red blood cells, but mainly in immunology we focus on the white blood cells because they're the ones that do the job of it. So in your bone marrow is where they are born, right? And it's where they develop these different receptors. So each B cell here, notice they're denoting its antibody, its B cell receptor, with a different color. And this is to signify, as we said, that each B cell is unique, right? Each B cell that's generated in the bone marrow is unique for a specific antigen that it doesn't even know about yet, right? It hasn't even seen it yet. And these are going to travel to our secondary lymphoid organs, right? So what are some examples of secondary lymphoid organs where these B cells will live and wait out to come and encounter with antigen? There's some sites on our body that have large collections of lymphocytes, B cells, T cells, and macrophages, and dendritic cells that are going to travel there. Our lymph nodes, right, all throughout our body, and then the specialized ones in our throat are what?
Our tonsils.
Tonsils, right? So remember, you have the palatine tonsils, and you have the one in the back of your nose, your nasopharyngeal, right? Your adenoids is the common name for it. All your mucus-associated lymphoid tissue, right? That's what we're talking about when we say mucus-associated. It's associated with the mucus layer, but lymphoid tissue meaning that they're B cells and T cells, right, that are able to produce antibodies and stimulate B cells to produce antibodies. In your appendix, in your spleen, right, and all your lymph nodes all throughout your body. But your B cells are born in the primary lymphoid organ, which is your bone marrow. And then T cells, remember, they're named after where they have to travel to for after they're born, their last bit of education of self, between self and non-self. What organ is that near your heart? Your thymus, right? So the same thing for T cells, right? Imagine their little T cell receptor with just one antigen binding site. They're all unique as well, okay? So then they talk about when it comes in contact with antigen, okay? So it actually binds to the antigen binding sites on those antibodies on the surface of the B cell. Remember, this is class IgM, right? That's on them. So this particular B cell, you could say, kind of won the lottery, right? But this is one B cell, right? Out of, thousands or millions. Is that enough to fight the fight? No, we need hundreds of these guys. So what happens now is that they're going to become stimulated. And notice later on they say sometimes that T cell, helper T cell, is going to stimulate this B cell. Okay? Sometimes it can do it on its own. It depends on the antigen. When we talk about clonal selection, so this guy's been selected, right? He's the one. He matches up. Clonal refers to that now we're going to make copies, right? A clone is an exact copy, right? So we're going to make exact copies of this B cell. Lots of them. 1,000 plus. Okay? And they're even going to travel throughout the body. The main thing that a B cell does is make antibodies, right? So it needs to differentiate now so that it will secrete them, right? So it goes through a process, it's been activated, right? And now we're going to produce lots of them. Some of those are going to differentiate into what we call a plasma cell, right? Think about the cytoplasm getting really big. It's actually the rough endoplasmic reticulum gets huge because you're producing a lot of protein that's going to be secreted. That's what antibodies are, they're proteins. Right? So it's making, secreting, and I read somewhere recently that, remember I told you guys about the amount of mucus you produce is like crazy amount? Well, I saw a reference to how much IgA you produce, so imagine how much of that antibody going into your secretion. 5 to 15 grams a day. That's a lot when you're talking about something molecular, something this small, right? So I'll try to bring in some type of reference for us a little bit later to help you fathom how much this is. So lots of antibodies. I mean, these guys are pumping this out, millions in minutes, okay? Some of them, though, are not going to differentiate into B cells, plasma cells that secrete antibody, They're going to become what's called memory B cells. So they look just like the B cell that was activated, right? They have that particular receptor. But now that we have more of them in our system, because it has seen antigen, the next time an antigen comes in, it's very quickly, hopefully, going to come in contact with one of these memory B cells. And our response is going to be much faster, right? And so, anybody watch the show Scorpion? It's one of my new shows that I've kind of been watching. And they very disappointed me. I got a couple watchers.
No, we actually talked about it in our speech class the other day.
Oh, did you?
It's really cool, yeah. It was funny.
Yeah, so I was really disappointed in them, though, because they did something that's not possible.
Really?
Well, yeah. I was like on the verge of writing them a note. As a scientist, I'm very upset, but then I was like, oh, perfect teaching moment. So when we talk about this process, how long do you guys think this takes?
Seconds.
Yeah, right? But it could take up to 14 days, the very first time you're exposed, and finding that one B cell, right, that matches up. And are you going to find just one B cell probably? No, because remember, antigens are quite complex. So there might be a couple of B cells that are going to get selected, right, that'll bind to that same virus or that same antigen. Still with me? Because remember, antibodies bind very specifically to little pieces, epitopes, on an antigen. Okay, so we may have several B cells that are selected, but this can take 7 to 14 days, typically around 14 days, to where you have the detectable levels of antibody. Okay, and so in the show, the guy got bit by a snake, a venomous snake, and you can inject that toxin, that antigen, right, into another organism, and especially if you do it at a low level, they'll produce antibodies against it. But how long is it going to take, y'all? Days. We're talking about days. They seem in the show that they could do it in minutes. And it drove me crazy, right? So the guy gets bit by a snake, they go and get this snake, they get a ferret, they take the snake venom from the snake and they inject it into the ferret, and somehow, minutes to maybe an hour later, if that, they're taking the blood out of this ferret to spin it to be able to take the liquid phase that has the proteins in it that would have the antibodies in it. There's no way, y'all, it happens that fast. No way, right? So I was very upset, right? As you can imagine, right? I'm like, no. I mean, it was great drama, right? But very bad science. The science behind it was true, though. You can do that. The problem was the time frame. And of course, we're talking about an hour-long show, 40 minutes without the commercials, right? So, in a snake habitat like that, you can check, Miss Patty, if you need to.
Oh, okay. Pam, thank you.
You're welcome. Pam, sorry. I knew it was a P, I'm sorry.
Miss Patty, you're right. Miss Patty, that's right.
Oh, it is Patty, okay. Now I lost my train of thought, but I was fine a second ago. So, what was I saying? Help me out.
You were talking about the right science, wrong amount of time.
Right, wrong amount of time. So the other thing with that is that, they go to this particular center to get this particular snake, right? A center like that, a reptile center or something like that, they're going to have anti-venom or anti-serum antibodies against that toxin on hand. So, if anything, they should have just gone and stolen the anti-serum from the place instead of, the snake. So, in this process, right, it takes time, right? And this is really going to vary. And this is the kind of scary thing when it comes to the adaptive immune response. Because if this doesn't kick in fast enough, right, the organism, the virus, the toxin, whatever it is, could potentially overwhelm your system and kill you before this kicks in. And so since we've learned about this system, we exploit this with vaccination, right? Where we can give you an antigen ahead of time and get you to produce antibodies and the very precious memory B cells, right? Because having more B cells that recognize that specific antigen are going to make this process go so much faster, right? Because it does have this memory component.
Well, then I don't understand how people can get allergies later on in life. Like how?
So your system is always, so she asked, I don't understand how people can get allergies later in life. So it has to do multifaceted. Your body is continually changing. Also, what environment you're exposed to changes, right? So you may not have been exposed to it so much, and now you're being exposed to it, which is activating this system.
Like now I'm allergic to the seasoning in crawfish. I always ate crawfish.
Right.
Now it's like, can't even eat crawfish anymore.
Right.
Or like whatever, like in seafood seasoning.
Girl, just change the bread.
I have to bring, no lie, I bring allergy medicine with me everywhere.
Yeah. You don't know. Yeah. And that's the thing is like, some people are more predisposed to allergies than others. And we're going to talk about allergy, but it's this same system, right? Where your body is recognizing it as foreign. It's producing these antibodies against it, right? And you're producing even more memory cells. So the more and more you're exposed to it, the greater and greater the response is. In this case, it's not a response you really want, right? So that's why it can be severely life-threatening when it comes to some food allergies too, because think about where these things are going. They're all going all throughout your body because you've ingested it, right? And it goes into your bloodstream.
My throat closes up and everything.
Yeah, oof, not good.
No, I'm not, I did 2 weeks ago. It's okay.
It's all good now. I'm thankfully still alive.
Crawfish, I have my medicine.
Yeah, but think about it, each time you're exposed, your reaction is going to be greater and greater. So stop. I know, it's tough. So as this is just a schematic, right? So it kind of lays it all out. And as you guys are studying, if drawing helps you, draw, right? No one else is going to see these. I did a lot of drawing when I was learning immunology, right? To help, visualize out the steps. So this is a good diagram for that. Again, driving home the point that they come from those stem cells in your bone marrow, right? And each B cell, when it's born, it's going to be unique, right? It's a cute little B cell baby. Has its very own antibody, right, on its surface. So again, denoted by different shapes and colors here, right? And even different letter designations to denote that each B cell is a unique when it's born. Okay. It isn't until it encounters antigen and then is stimulated, if need be, by a helper T cell, that it's going to then proliferate, right, upon activation. And as you saw in that animation, remember, they talk to each other. They don't only basically shake hands by joining up by those receptors, but then remember the cytokines, the interleukins, right? They're communicating, yes, I recognize you. Yes, this is what you should do next, right? We need more of you. So this clonal selection, so you've been selected, you're going to expand lots of these. Some of them are going to differentiate into plasma cells when we talk about B cells to actually secrete the antibodies. And then others are going to stick around as memory B cells in case we see this bad guy again, right? We don't want to forget what he looks like. Because chances are you're probably going to see it again, right? Somehow our bodies figured this out a long time ago. We might see this again. We might want to remember this bad guy. So actually I want to go back up to this original slide that I kind of skimmed over earlier. So B cells and T cells, although we focused on the B cells in the animation and in our discussion so far, this is true for T cells as well, okay? They're going to have their own specific T cell receptor, and they have to be activated, except they communicate, remember, with antigen presenting cells, not with antigen directly, right? So we'll look at that. They're able to recognize functionally limitless variety of antigens. When you read that, you're like, oh, but we're talking about the population of B cells and T cells that you have, right? Because each one of them is different and unique. Within your body, you have the potential of hopefully recognizing just about any antigens out there. And you're continually making these B cells, right? And continually making T cells. And you're going to make them even more when your body recognizes that it has an infection, right? It's going to be stimulated. Those colony stimulating cytokines, right, the colony stimulating factors are going to stimulate that bone marrow to produce more B cells and T cells and macrophages and whatever it is it needs. So as it says, each cell, though, only recognizes and responds to a single epitope. We're talking about a single little piece on an antigen, right, that antibody is actually going to bind to. Same holds true for the T cell receptor. It only recognizes a little piece. It's a pretty amazing system when you think about it. The diversity that we have is a random system. I almost equate it to playing the lottery, right? So when our B cells are born, right, that jumbling up that they do to choose their B cell receptor is a random process, right? It's literally like randomly picking lottery numbers, right? But they have a set amount of DNA to play around with, okay, and organize it. But there's a limitless number of combinations that you could do jumbling up that DNA. So then they sit around in those lymphoid organs, right? In the spleen, your tonsils, and the mucus-associated and skin-associated lymphoid tissue, your molten salt, right? For some macrophage or dendritic cell that might bring something in, or just the lymph itself traveling through a lymph node might have some antigens in it, right? That'll bring them to that area. That specificity of the antigen receptors governs recognition, right? So it has to match up. But that's not just it, right? So sometimes, just matching up is not enough. Although polysaccharides and proteins that can bind, you notice how in that animation, the antibodies kind of all clustered together. They call that capping. Remember before I talked about string response? So string response is where, say, like lipopolysaccharide, that's a really good, it's a sugar component. It's a highly repetitive molecule. So lots of it can bind to lots of antibodies on the surface of a B cell. So it almost creates like a string. And it can help form that cap, and that can be enough to stimulate that B cell to go forward without a T cell helping it out. Okay, so now it's going to proliferate, right? It's going to produce plasma cells that secrete antibodies and memory B cells. But usually it needs confirmation from another cell type. Now for a B cell, that would be a helper T cell. For a T cell, right, activation is always going to be by antigen-presenting cells, whether it be our own natural cells or antigen-presenting cells like the macrophage and the dendritic cells. Right, then they're going to multiply, right, specifically for the B cells. They'll leave, as I said, the lymphoid organs are going to migrate to tissues. They're going to travel throughout the body. They're going to go right to where the action is, right to where the antigens are, and produce antibodies right there as well. And then they go through a process we know as apoptosis when they're no longer needed. And what happens when they go through apoptosis? They're destroyed, they die. But unlike when death happens from injury or infection, it's not going to elicit the very common immunological response, which is inflammation, right? So when these cells die off, right, we're going to be ending the inflammation cycle, right? We've taken care of the bad guy. We wouldn't eliminate them unless their job was done, right? We don't want to maintain them, right? These large numbers of cells, that's costly to the body, right? And this is also why you're exhausted when you're sick. Think about all these cells and proteins that your body is producing. That takes a lot of energy, which is why rest is paramount when you're sick, right? Because you want your body to have the energy necessary to fight. You don't want to be using it to do other things. You want your immune system to get most of your energy. And anyone who's been sick, which is everyone, right, knows what that zap of loss of energy feels like, right? And this is why, right? Your body is working really hard to save your ass. Right? So lymphocytes, right, when they're first born, they have an antigen receptor, but they haven't encountered it. They were what we call naive, right? They don't know about that there's a bad guy out there. Once they're activated, once they've encountered antigen, whether it's been presented to them or they come in contact with it, they're going to be able to proliferate. They've become activated, right? They have bound to and found their specific antigen. And then depending on your cells, right, they can differentiate to what we call effector cells. They can cause effective change, right? They can do something. So all of them are able to produce cytokines, right? They could communicate their actions to other cells, right? And only effector cells only come from activated cells, right? So this is our progression, right? Naive, I've never seen it before, right? I become activated because I've bound to antigen. I recognize it. I may even been stimulated. Now, some of us are going to differentiate into effector cells. When we talk about B cells, that's going to be plasma cells that actually secrete antibodies, right? So you go from B cell to a plasma cell. For T cells, right, you're going to become helper T cells or cytotoxic T cells. Helper T cells, as the name implies, are going to help out other cells like the B cells or the macrophages. Cytotoxic T cells, cytotoxic in its name kind of lets you know what it does, right? It goes out and kills cells, right? Cytotoxic. Cyto referring to cell, toxic, death. And then for B cells and T cells, we're always going to produce memory cells, right? Whenever we've been activated, We've made effector cells, and these are long-lived descendants of those activated B cells and T cells. And as I said, these are the ones that really help us out the second time you come in contact with something. The first time you may get sick, and I mean, you're down and out, you're tired, you feel like crap, but you win the battle and you live, right? The second time you come in contact with it, you might not even know you did, because your body is that fast at responding to it and clearing it up quick enough that you don't even know that you had a quote-unquote infection, an invasion that happened. That's how impressive it can be the second time around. So what role do our helper T cells play in B cell activation, right? They're giving them that signal, okay, I recognize, go forward. What's the similarities and differences between primary and secondary response? So primary is the first time we ever see it, right? Secondary and subsequent after that. We have what that's going to help us out? What's going to help us out that second time? What is the great thing about this system? You have what cells? Memory cells, right? The memory cells, right? But the first time, do you have memory cells? No, right? That's the difference. That's the key difference there between your primary and any secondary response, is that the first time you don't have those memory cells. You don't have those predetermined ones that recognize this antigen. And so the time frame is very different. But is the process the same? Yeah, we're still going to get memory cells activated. They're still going to proliferate. Some of them are still going to turn into plasma cells and secrete antibodies. Others will turn into more memory cells, right? So you'll have even more. Same thing with the T cells, right? You're going to get those memory cells. You're going to get more effector cells, whether it be helper T cells or cytotoxic, or maybe even both, depending on what's going on. Now, T-dependent and T-independent antigens, right? And this is really related to B cell activation. So what's the difference? It's pretty simple, right? T-dependent, they have to have T cells helping them out to activate them. T-independent, as the name implies, do they need the T cells? No, right? B cells can do it all on their own. And I have no clock today. I left my watch at home. Here we go. Thank you. So this process, right, of going B cell to plasma cell to secreting, as we saw on the other animation, here it comes in contact with an antigen, specifically at a little piece. It's going to bring it inside. It's going to then present it outward. Okay? A helper T cell can interact with it. Remember, T cells can only interact with antigen-presenting cells. A B cell, in this case, is considered an antigen-presenting cell. Okay? It's presenting antigen. And remember, it's a special class of molecule that it uses. It's MHC class 2. This stands for major histocompatibility complex. And where it got its name is this is the ones that create problems when we try to do tissue and organ transplants. These are what the body doesn't recognize and therefore attacks your cells, right? Because if your immune system, you get them from someone else, their DNA is different, their MHC is a little different. So that's where the major, right, it's a major player. Histocompatibility, as in recognizing, right, being compatible at the histological level, at the tissue level, right? Histology refers to tissues. Okay, so there's two different classes of these molecules. One is found on your antigen-presenting cells. It really is the first class that's found on all of your cells, class 1, that creates the major problem with organ and tissue transplants. Because this is really communication within your own system. So notice that the cell is presenting, the T cell is recognizing it, right? And it's sending signals, cytokine signals, and it's activating that B cell. That B cell sometimes is not going to be activated unless a T cell recognizes. So again, look at the time frame that this may need to happen if this is the first time we saw the antigen, right? And we found, 4 or 5 B cells that recognize this particular antigen. But then they got to wait for their helper T cell buddy to say, oh yeah, I recognize it too. Okay, and how did that T cell get activated? Well, it probably came in contact with a dendritic cell or a macrophage that told it, right, oh yeah, this is the bad guy, he's here, you recognize, okay, go tell all your friends. And they're going to proliferate so there's more of them. So all of this takes time, right? This is multiple steps in the process in order for us to get to where we're actually secreting antibodies. So as I said, there's no exact timetable written here because it's going to vary, right? But in general, you're going to have this lag period, this period of 10 to 12, lots of different ones say different things, right? The average, they say, is around 7 to 14. That's the average range, right? Where they actually become detectable in your blood, right? We're going to pull the blood out of you, right? After you've been exposed to an antigen for the very first time, right? You're going to have this lag period. And then notice the first antibody we said you produce is IgM, and then commonly your body goes through what's called a class switch. And in the core parts of your body, that switch is going to be usually to G. Where when we talk about your mucous membranes, remember what's the predominant antibody in your mucus? What class of antibody? What's in your mucus? IgA, right? And so commonly those B cells are going to switch to A in those particular regions. And why are they going to do that? Signaling in that area, right? Cytokines and communication that happens that says, you need to make IgA, right? And then those of us that are more predisposed to allergy in certain regions of our body, we're producing way too much Ig. Which one's associated with allergy? Erica hates it. IgE. Right? You can remember E for Erica is not very fond of IgEs. Got way too many. Okay? But the most common is going to be IgM to IgG is your most common switch. But, in particular areas, especially associated with mucus, you're going to switch to making IgA, or you're going to be switching to IgE. And most of us aren't too happy about that switch. Oh, I'm getting ahead of myself. So notice, and this is concentration. And of course, as we eliminate the pathogen, it's going to go down, right? That concentration is going to go down. But you're going to have a certain level that's going to stick around. And especially with IgG, this really is a long-lasting antibody. The second time you get exposed, notice literally, right? Almost no lag. We're talking about, it could be the potential of hours, right? That you start producing IgM and very quickly switch to extremely high levels of IgG. Why is this? What cells are responsible for this very quick response? The memory cells, right? Because you just, in general, have more of them, right? So you're going to respond that much faster. So that's it for me today, right? I see you guys packing up on me. That can be it. That can be it for today. All right. So we'll go into more details next time. So remember, your best bet is to review as close as you can after lecture, go over stuff. As I said, I clicked